Welcome to the course on basic genetics. As stated in my earlier lectures, three of us are sharing this course: Dr. Richa Arya, Professor Madhu Gavaldas Tapadia, and myself Jagat Kumar Roy. We are from Department of Zoology, Banaras Hindu University. I am a retired person now. This is week 10 course and fifth lecture, Fluorescence in situ hybridization or FISH. We have seen G-banded chromosomes. G-bands provided easy identification of each chromosome very accurately. So far we are quite familiar with G-band. Now the question is, can we go to the molecular level? Can we do some banding which is at molecular level? Can we have more precision? These are the things. With this we come to the fluorescence in situ hybridization. When we say fluorescence, fluorescence means something that fluoresces or some chemical that absorbs light at non-visible wavelength and emits light in the visible wavelength. So that is fluorescence. That means a fluorochrome will be used which will absorb light, say, in the UV range, and which will emit light in the visible range. That is what we will be able to see. Now the other two terms are in situ hybridization. In situ means in its original location, and hybridization means something which we are allowing to detect another thing. That means if we talk in terms of DNA, we have chromosome, in the chromosome we have DNA, or we have isolated DNA and we have DNA. Now if suppose we have a small part of a gene that we call as a probe and which we want to use to search the gene of our interest, then the technique used will be hybridizing this probe to the main DNA and identifying the region. So what is a probe? Once again, probe is a short single-stranded fragment. This is a short single-stranded fragment of DNA. When we say single-stranded fragment of DNA, it is not necessary that probe has to be single-stranded. A double-stranded DNA denatured to make single-stranded is also fine. It can be an RNA also that can identify a specific target sequence by complementary base pairing. So this is what is a probe. That means, for example, if suppose I have a cap of a pen in my hand and several pens are there, how I'll identify the pen of our interest? By using the cap and trying with different pens. Wherever it fits, I'll say this is the pen I was looking for. Exactly same thing will be here. Now probe is a molecule. This is a small DNA molecule. How we will identify where the probe is? That means probe has to be labeled. Labeled means this has to be tagged with something, a fluorochrome or some kind of molecule which will give a color reaction or such kind of thing. There are different ways by which the probe can be labeled. See, this is a double-stranded DNA. This is a double-stranded DNA. And with the DNAs, there can be random cut, and some of the places the bases can be removed. It can be done with DNAs. It may not be done with DNAs by itself also. The process of isolation of DNA has lot of nicks. And if we use DNA polymerase, the complete DNA polymerase, this can widen the neck, because DNA polymerase 1 I am talking, this has two units. One is the clino fragment that has 5' to 3' polymerization activity and 3' to 5' exonuclease activity. This is called proofreading activity. That means the first work is polymerization. If there is something wrong, this will come back, remove the thing and again will polymerize. So, that is what is the clino fragment. If you take the entire pol 1, DNA polymerase 1, this has 5' to 3' nicking activity also, if 5' to 3' polymerization activity is not being given at this moment or not being allowed at this moment. For example, you have taken a double stranded DNA, almost double stranded DNA with some nicks. The DNA polymerase will not find it easy to go for 5' to 3' direction polymerization. Rather, it will be easy for the molecule to make 5' to 3' exonuclease activity, so that some part is removed and then 5' to 3' polymerization activity. So, this is a neck and then the neck is widened and then this is polymerized. This is called neck translation. So, this is the neck translation, that there are necks and here you are using dig DUTP. This is a labeled deoxy-UTP and this is dig means this has a label with it. So, this is fluorescence, this is fluorescently labeled and this will get incorporated if DNA synthesis is there. So, when DNA synthesis is occurring, this got into the double stranded DNA. So, this DNA has now got labeled. If you denature it, this becomes single stranded and that is ready as a probe. The second way is by random priming. So, here DNA is denatured, then random hexanucleotide primers are added and then the clino subunit, as I discussed, that along with the DCTP, DATP, DTTP, DGTP plus label. So, this will polymerize. While doing this polymerization, this labeled deoxyribonucleotide is being taken and thus the probe is labeled. So, now the probe is ready. Now, we have to do hybridization. Where hybridization has to be done? This has to be done on a cell. On a cell means in the DNA of a cell, or on chromosome, that is chromosomal DNA that is present within the cell, within the nucleus, or tissue section on a slide, where again the moment we say in situ hybridization, the hybridization has to be done with RNA or the DNA. So, this is denatured DNA that is there, means if we are taking cell, we have denatured it with formamide or with temperature. So, the DNA inside the nucleus is now in a single stranded condition. Now, the probe is allowed to hybridize. So, this is the probe, this is hybridizing with this. Wherever it hybridizes, this will stay. If it stays, now this DNA is now labeled. This is now labeled. And if we use antibodies against this, so we have now antibodies against this. This has captured this and this antibody is having high fluorescence. This has fluorescence. So, result is Now this will start fluorescing wherever the hybridization is. So now I show you a picture. This is RNA in situ hybridization. Suppose aim is to see where a particular gene is being synthesized or being transcribed in a cell. Transcription always occurs inside the nucleus, but the transcript will go and will be placed where the translation is required. So this is the nucleus of the cell. The transcription occurred here, then RNAs came out and they are now settled in the cytoplasm, and that we are seeing by in situ hybridization. This is the pattern of a given gene, and these cells are HeLa cells. Okay. Can we do this? This is the earlier one, just now we showed, that is RNA in situ hybridization. That means the expression of a gene is being traced. Now, can the hybridization be done on chromosome to localize, let us say, a gene? Yes. So a probe has to be specific for a given gene. Here in this, 2 gene probes are being used. One is with green color, one is with red color. That means wherever this red colored gene is present, a gene named, let us say, ABL, wherever that is present, this will be shown on the chromosome. The other gene, let us say gene named BCR, wherever in whichever chromosome it is present, it will be shown like this. Now, if by chance there is a translocation, that a part of this chromosome and a part of this chromosome has come together, you can see this has come together. So this way you can pinpoint where the gene is present on the chromosome, and if there are some kind of anomaly because of that, these 2 genes have come together. So this is one method of identification of a gene on chromosome. Now the techniques have advanced further, and chromosomes can be painted by in situ hybridization. That means we have to use specific kind of protein or specific kind of means gene for specific kind of protein or specific kind of molecule, and they are hybridized. So you can see some of the chromosomes are red, some of the chromosomes are with yellow, and this is because of hybridization with more than one kind of probe on the DNA, on the chromosome, and they can be seen. In this also, if you see, this one is the normal chromosome. You can see chromosome number 1, it is like this, chromosome number 1 is like this. In the other one also, you have chromosome 1 like here, chromosome 1 here. Chromosome 2 looks like this, chromosome 2 looks like this. So this is chromosome 2, this is chromosome 2. Chromosome 4 is like this. So here chromosome 4 has an anomaly. You have part of this chromosome, part of this red that has come, that is a translocation from somewhere else. So this way, by painting the chromosome, you can find anomaly. Use of a combination of probe to paint chromosome differentially. Now your aim immediately, when you can do this, your aim may be to try different chromosome with different color. That means you have to have the specific probe that can hybridize with a particular chromosome, another specific probe that can hybridize with another specific chromosome, another probe that will hybridize with another specific chromosome, and each chromosome and each probe will have the different fluorochrome with it. So One will be fluorescing pink, another will be fluorescing yellow, another will be fluorescing red, another will be fluorescing green, and so on. So, this way you can paint the different chromosome, and the identification now becomes easy. If you say chromosome 1, I have given color green. So, the green color chromosome will be chromosome number 1, like that. So, this is the way the chromosome painting is very much useful. I will show you. We were talking so much about karyotype. Is human karyotyping possible with a combination of probes? Yes, it is possible, because we can paint the different chromosomes. Once again, when we are painting, that means we are using different probes, several different probes which are hybridizing to one chromosome or might be hybridizing to other chromosome. So, this way you can see chromosome 1 is having certain color. So many different probes have hybridized with chromosome 1. Many different probes have hybridized with chromosome 2, with chromosome 3. Chromosome 4 is having a different other kind of probe that has hybridized. Chromosome number 5 is different. So, this way you can do a karyotype very easily. Now, the question is that if we can do these at molecular level, if we can do karyotyping, is it that such kind of karyotyping will be much better than G banding, much better than other banding? Can we replace them? That is the next question. Answer to this is not at all, because every technique will have its own limitation. And when we talk about, I said that G banding, the nearly 100 years, the G banding is being followed, if not 100 years, 80 years or so, G banding is being followed. So, the G banding is having its own advantage, and if you see here, it is not only 1, 2, 3, 4 bands. Each long chromosome of G banded chromosome will have several bands. In high resolution, this will have several subbands. Is it possible with this? For single gene identification, this is very much possible. For 2 gene identification, this is very much possible. But for karyotyping, I will say that we should go back to G banding only, or G banding, Q banding, those kind of classical bandings for the identifying the chromosome, for identifying the different chromosome regions. I tell you that in earlier days, when so much neither molecular biology was known, nor the photography method was very well developed, if you see the precision of certain kind of banding, certain kind of photography, they are much sharper, much clearer than the present day. So, that means we might have lost some details, we might have gained something else. Always one has to look for what for we are looking, and that has to be very clear. So, with this I come to the discussion. Can we paint all the different chromosomes with different color? Answer to this is with all probability yes, provided we have so many different probes with us. So many different probes which can specifically hybridize to one chromosome, not to other. If we get such kind of probe, then yes. Is by fish chromosome identification is more accurate than G banding? I will say once again that if it is single gene identification, as I will go back once, if it is this kind of single gene identification, you can see here, you can see here, then fish is much better than any banding or any other kind of technique. But if it is full chromosome karyotyping, the G-banding karyotyping will be much better. Can these be visualized by a bright field microscope also? For these, for fluorescence in situ hybridization, you will require fluorescence microscope, where the light source will have the ultraviolet range of light, and we will be visualizing the visible light in range. So, that means bright field microscopy will not be possible. Is this the time to replace all conventional chromosome banding method with molecular hybridization? I did say no, that this is not. Always one has to be very clear on aim that what we want. Do we want identification of all the different regions of a chromosome? Then no. If you want identification of one chromosome, yes. If we want identification of a gene within the chromosome, molecular technique will be much better. So, these are the things, these are the discussion points, and we should discuss in detail whenever time permits. I thank you.